Welcome to this UCL Lunch Hour lecture marking World Alzheimer's Month on the future of dementia research. My name is Fiona Ducoteur. I'm the Chief Scientific Officer of the Alzheimer's Research UK Drug Discovery Institute here at UCL. It's my honour and pleasure to introduce today's speaker, Professor Selina Wray. Professor Selina Wray is a professor of molecular neuroscience in the Department of Neurodegenerative Disease, UCL Queen's Square Institute of Neurology. Her research focuses on understanding the molecular mechanisms of Alzheimer's disease and frontotemporal dementia using patient-derived stem cell models. Her work sheds light on the earliest changes in the brain while enhancing our understanding of disease mechanisms, and this aids us in the development of novel therapeutics. Professor Wray is also a passionate advocate for public engagement and has received multiple awards for both her research and her outreach, including the Alzheimer's Research UK David Haig Early Career Investigator of the Year Award and the Suffrage Science Award for Life Sciences. Before we begin, I wanted to let you know that we will have some time at the end of the lecture for questions, and these can be submitted at any point during the talk by going to Slido and entering the event code #WAM, which stands for World Alzheimer's Month. I'll now hand over to Selina for her talk.
Thank you so much, Fiona, and good afternoon, everyone. I will just share my slides if you bear with me 1 second. Fantastic. Hopefully everyone can see those. Wonderful. So I'm really excited to spend the next 40 minutes or so talking to you a little bit about dementia research, particularly research that has been happening at UCL and some of the research that has been happening in my own group, and how that is helping us move closer towards treatments for diseases such as Alzheimer's disease. And although the title of the talk is The Future of Dementia, as I was preparing the slides, I realized maybe a more accurate title would be, to say the past, present, and future of dementia, because to really understand where we are going and why this is such an exciting time to be involved in dementia research, I think we need to reflect a little bit and understand how we arrived at where we are now. And so hopefully I'll be able to talk to you a little bit about that over the next 40 minutes. And as I mentioned, I will also talk a little bit about the work that we are doing in my own lab. So I've given myself a subtitle of Building Brains in a Dish to Understand Dementia. And you can see, hopefully, as my background and also on screen, a really beautiful image, in my opinion, beautiful. And if you stay tuned towards the second half of the talk, I can really tell you a little bit more about what this shows and why it's such a powerful tool for us. So to give an overview of what I hope to discuss with you during this session, I wanted to start first of all by providing a little bit of context. Why is dementia such an important area to be working in? Why should we all be interested and engaged with dementia research? But also to clarify a few kind of terms that we use around dementia and what specifically they mean. I then would like to briefly walk us through a history of Alzheimer's research, just to bring everyone up to speed about where we are now, what's happening in the world with diagnosis, but also with treatments. And as I do so, I'll talk about some of the challenges that we've faced along the way. And I really would like to tap into that a little bit more to help everyone understand why this is, although it's important, also such a challenging area to be working in, but also share some excitement of how stem cells can help us and where we are heading in the future. So as I mentioned, I will first of all start with hopefully providing some clarity about some of the words we use when we talk about dementia. So I speak about dementia research quite often to various groups of people. And one of the things that I am most commonly asked is, well, what is the difference between dementia and Alzheimer's disease? Are they the same thing, or are they different things? And this is a really good question, and it's something that actually I'm quite guilty of sometimes, is using those terms interchangeably when they actually mean quite different things. So the word dementia is an umbrella term, and it really describes a set of symptoms. Now, these symptoms include, most people would most commonly think of memory problems associated with dementia, and that is indeed one of the most common symptoms. But there can be other symptoms that come under this umbrella term, such as confusion, difficulty with navigation. So people may suddenly start to have trouble finding their way on a route that they've walked every day for the past 20 years. It can be associated with personality changes. So people may become uncharacteristically angry or bad-tempered, for example. And these symptoms can be caused by diseases of the brain. And the most common disease which causes dementia is Alzheimer's disease. This is sometimes why we use the terms interchangeably. And actually, for the majority of my talk today, I will be focused on Alzheimer's disease, but I really think it's important to highlight some of the other causes of dementia, which some of you may have heard of, some of you may have not. But for example, frontotemporal dementia, this is named because it affects the front part of the brain. This is quite commonly associated with personality changes rather than memory loss. It can also be associated with our ability to use and understand language. And actually, although this is rarer than Alzheimer's disease, many of you may have heard of it recently because it's sadly what Bruce Willis is living with at the moment. And so we're starting to see an increase in awareness of FTD in the news. Other diseases can include dementia with Lewy bodies and also vascular dementia. So for example, dementia that can arise following or as a consequence of stroke. But Alzheimer's disease is by far the most common cause of dementia. Now, this is really an important point because for many years there was a misconception around dementia being something that's inevitable as we get older. And this is actually not the case. If we acknowledge that dementia is caused by diseases in the brain, that gives us optimism because it means that we can understand those diseases through research and we can develop treatments to stop those diseases or slow them down. I also wanted to highlight that dementia is incredibly common. And these are some facts and figures that I took from the Dementia Statistics website, which is hosted by Alzheimer's Research UK. There are almost 1 million people living in the UK who are living with dementia at the moment. So 982,000. And this means that 1 in 2 of us will be affected by dementia in our lifetime, whether we are living with the condition ourselves or caring and supporting a loved one who has the condition. We also know it disproportionately affects women, and we don't fully understand the reasons for this yet, but over half of the people living with dementia are women, and actually women are much more likely to take on caring responsibilities. So it does have a disproportionate effect on women. Now, having a condition which is so common, but not really having treatments that can slow down or stop the progression of the disease means that dementia is also very expensive. And the cost of dementia to the UK economy is actually more than stroke, cancer, and cardiovascular heart disease combined. So this is really a huge cost. And as you can see on the graph here, the cost takes several forms. Economic costs, social care, and also unpaid care also all contribute to the cost of dementia to the UK. And of course, reducing dementia to numbers in this way really doesn't take into account the personal cost for people who are supporting a loved one living with one of these conditions. So although this is quite a maybe difficult start to a talk, I hope it does show you that this is really an urgent public health need. And we really need research to improve diagnosis, to understand the different conditions that cause dementia so that we can develop those new treatments. And actually something that would even slow down the progression of the disease by maybe 5 years would have a huge impact on the quality of life and the facts and figures I've shared with you just now. So what are we doing about dementia? And just to introduce myself a little bit, as you heard, I'm a professor of molecular neuroscience. I'll talk about that in a second. I run a lab doing lab-based research, and I'm based at UCL Queen Square Institute of Neurology, which is really a remarkable place. One of the things that makes it unique and makes it such a fantastic place to do dementia research is the co-location of the clinical and preclinical work. So the image on your screen here is Queen Square, which is just by King's Cross in London. The red brick building is the National Hospital for Neurology and Neurosurgery. So this is really one of the kind of leading neurology hospitals in the UK. And many people with dementia will be seen by the neurologists in this hospital and will choose to participate in research. So clinical research, for example, through donating blood samples or donating things like CSF. But next door to the hospital is this concrete building, which is the Institute of Neurology. And this is the research institute that partners with the hospital to really drive forward research into neurological conditions. Now, what's special about the institute is we really have a single mission, which is to do discovery research in the lab and translate that research into diagnostic or treatment that will improve the life of neurological disease. And I've been really privileged to work in this setting because, as I will show you, the way we really make progress in these conditions is by having the clinical and the lab-based research working together and integrated to make faster progress. And so I'm a molecular neuroscientist. That's a very fancy way of saying that my own research is focused on understanding the small stuff. So specifically, what I mean by that is we work on DNA and proteins. These are the smallest building blocks of the cells of our body. And in the brain, we're really interested in two types of cells. I'll tell you more about them in a second, but neurons and then a different type of cell called glial cells. As I mentioned, these cells come together to make our organs, so the brain being the one that we are most interested in, and ultimately these come together to make us. And the reason I showed this schematic is because I think it makes it easy to appreciate that if something goes wrong at the very molecular level, the consequences of that will be felt at each increasing level of complexity. So if you have a change in your DNA or you have a protein in your cell that no longer functions properly, that will cause the cells to stop working as efficiently, which impacts how our organs can function and would ultimately lead to symptoms such as memory loss or problems with confusion or movement, for example. This also makes it easy to see, I think, that if we want to treat these diseases, we need to focus those treatments here. We do want to have treatments that will improve and alleviate people's symptoms, but if we want to stop or slow down the disease, we really need to intervene at this molecular level. So in Alzheimer's disease, I thought it would be useful to just talk a little bit first about what the Alzheimer's disease brain looks like. And on the left-hand side of the screen, you can see a picture of somebody's brain who has lived with Alzheimer's and come to the end stage of their disease compared to a healthy brain. And when I say healthy brain, I mean somebody who hasn't lived with Alzheimer's disease. And I think it's not necessary to be a neuropathologist to see that there's been a vast amount of change in the Alzheimer brain. So what you can see is there's been a huge amount of shrinkage of the brain. Actually, the brain of somebody who has lived with Alzheimer's weighs 1/3 less than a kind of intact brain that hasn't had this damage. And that loss of mass is caused because the cells of the brain in Alzheimer's will progressively die over time. So we call this a neurodegenerative disease, which means the cell type of the brain called the neurons degenerates over time. And it's that degeneration that will result in the symptoms. And you can see that here we have this, what we would call atrophy, these kind of holes in the brain, which would be previously occupied by neurons that are no longer there because they have succumbed to the disease. Now, if we were to look at this brain more closely under the microscope, we see two key pathologies which define the disease. And it was actually Alzheimer was the first to observe these, and it's why the disease went on to take his name. So on the right hand of the screen, you can see what these pathologies look like. In the upper panel, these kind of brown accumulations here are what we call amyloid plaques. And they're made of a small piece of a protein, which we call amyloid beta or A beta. And then in the bottom half of the screen, these flame-shaped inclusions, which are coloured in black, are what we call neurofibrillary tangles. And these are made of a protein called tau. Now, looking at the brain at post-mortem has been incredibly valuable to us in terms of understanding what defines Alzheimer's disease at the pathological level. But of course, one of the big challenges of working with post-mortem brain tissue, as I will come on to talk about later on, is we are really getting a single snapshot in time. And that snapshot is at the end of somebody's disease course. So although we can see the plaques and the tangles and the neuronal loss, what we cannot tell only by looking at postmortem tissue is the order in which these things happen. Did the plaques come first or the tangles come first? We also can't tell which is more important. Are the plaques causing the neurons to die? Is it the tangles? Is it both? Is it something else? And to start to put together a timeline of Alzheimer's disease, our progress has really been informed by genetic discoveries. So I wanted to first of all talk a little bit about the genetics of Alzheimer's and how that has informed where we are now as a field. And so I wanted to really mention somebody who's been a really inspirational figure in Alzheimer's research, and that's the lady you can see on the screen here. She's called Carol Jennings, and she wasn't a scientist by training, but for me, she's really one of the most influential and inspirational women in Alzheimer's research. And the reason for that is this is her family tree. And in the late 80s, she sent a handwritten version of this to John Hardy, who is a geneticist based at UCL, and Martin Rosser, who is a consultant neurologist. And she said, look, I think my family may be of interest to you. And actually, the reason her family was so interesting is everybody who is coloured in black on this family tree is somebody who developed Alzheimer's, but they developed it at a very early age, maybe in their 50s, and had a very aggressive form of disease. And through working with this family, John and Martin were able to identify the first gene that causes Alzheimer's. So Carol's family had something called autosomal dominant Alzheimer's, which means if your parent has Alzheimer's disease, you have a 50% chance of inheriting the gene that causes the disease. Now, this is very, very rare, but by studying these autosomal dominant families, we've been able to gain a huge amount of information about Alzheimer's disease progression. So before I talk a little bit about the genetic findings, I thought we could hear a little bit about it directly in the words of Carol and John. And so I'm going to play a short video that will just last about 3 minutes, where you can hear a little bit about it themselves.
The quest for medical treatments and cures isn't just about slides and microscopes and what can you do about the symptoms. It's about families, it's about individuals, it's about stories of people who suddenly begin to lose themselves in a way that's frightening for them and terribly painful for those who love them.
A lot of people would say that Alzheimer's disease research to some extent started with Carol's family.
All of them were between the ages of 54 and 58, and it was at that stage that our life changed, really.
We put an advert in 1985, 1986 in the Alzheimer's Society newsletter because we were interested in studying the genetics of Alzheimer's disease. And the very first letter I got was from Carol Jennings, and she had drawn her family tree, described her family tree.
The breakthrough itself came in 1991. We got a letter in January telling us they had identified a gene that they believed causes Alzheimer's disease.
When we found the mutations in amyloid in Carol's family, we knew that is where the disease started. It's been a dominant feature of all work on Alzheimer's disease. Carol's family has what's called autosomal dominant Alzheimer's disease, and that means that if your father or mother has the disease, you have a 50% chance of inheriting the gene, and if you get the gene, you're going to get the disease at a very similar age probably to your parent.
She's a shell of the woman she once was, but she's still my wife, and so she's still part of who I am. We may not be able to change the cards that life deals with us, but we can change the game we play with the cards we've got so that future generations don't have to carry the same pain.
So I hope that conveys some of the importance of Carol's family, but also more broadly, how indebted we are to people who choose to participate in research and share something of themselves to kind of advance our understanding of disease. And if anybody is interested in this, the video I showed is quite a few years old. There has actually been a wonderful documentary called The Jennings versus Alzheimer's, which I believe is available on BBC iPlayer and featuring many researchers around UCL who have worked with the family. And I would really recommend watching that for anybody who wants to understand a little bit more about autosomal dominant Alzheimer's disease and about their experience. So as I already mentioned, autosomal dominant Alzheimer's that is inherited in this way is incredibly rare. But because the pathologies, the plaques and the tangles are shared across Alzheimer's as a whole, it really can give us important insights which are relevant to Alzheimer's, whether it's autosomal dominant or the later onset, what we would call the sporadic form of the condition. And as you heard John say in the short video clip, the finding from Carol's family was that it's mutations in amyloid which drive the disease. And this led to the development of something we call the amyloid cascade hypothesis. And in a nutshell, what that means, if we think back to the pathology that I showed you with the plaques and the tangles and the neurodegeneration, it tells us that the plaques are happening first, because all of the genes that we know are linked to Alzheimer's disease, the autosomal dominant form of Alzheimer's, are linked to the generation of this amyloid beta peptide. And actually, there was an earlier example of this, which is trisomy 21. So the amyloid gene, APP, is located on chromosome 21, which means that individuals with Down syndrome have 3 copies of the gene. And as many of you may already know, for people with Down syndrome, they typically will go on to have a higher incidence of early onset Alzheimer's disease. So 2 points here, and one thing we may come back to in the questions or may mention later is just because amyloid is happening first, it does not mean that the tau is not important. It just means it happens later in the disease timeline. But also the reason this was such a critical piece of information is it suggests to us if the amyloid is happening first, if we block the amyloid, we will stop everything that comes downstream of that amyloid. And for this reason, the development of amyloid targeting therapies has really been the focus of a lot of drug development in Alzheimer's for the past kind of almost 40 years. But it's been incredibly challenging, and we've had a lot of drug trials that have failed. That narrative is starting to change, which I will show you in a few slides' time. But I think it's worth to pause and think about why has this been so challenging. And what we realize now is perhaps it's related to when does the disease start. So a lot of the early trials, to actually be a participant in one of those clinical trials, you had to have a diagnosis of Alzheimer's disease. And on the surface of it, that makes perfect sense. But actually, what we understand now is the disease is starting at the cellular level many years before somebody will develop their first symptoms. And this is a video that was kindly shared with me by one of my collaborators, Professor John Rohrer, who's at the Dementia Research Centre. And this is a series of MRI scans taken over a 5-year period from an individual who carries a causative gene for dementia. And what's really powerful about genetic dementia is we can follow people before they develop any symptoms. And what you're seeing here, if I just replay the clip, is over the 5-year period when these scans have been taken, we are seeing the brain shrinkage. We're seeing neuronal loss in real time. But during the duration of this clip, the person was not showing any symptoms of dementia. Now, why is that important? Well, it's telling us we need to intervene really very early in the disease course in order to have the best chance of our treatments being effective. And this is just another way of kind of showing that. This is a really famous figure in the field of Alzheimer's that we call Jack curves, but we essentially are showing that the amyloid and the tau changes in brain cells are happening maybe 2 decades. They start to happen 2 decades before someone develops their own symptoms. So this tells us 2 things. 1, we need to treat as early as possible, but 2, of course, the majority of people don't have these causative genes. And so it means we need ways that we can diagnose people early so we can give them treatments at the time when they were most likely to be effective. And recently, if anybody has kind of sees Alzheimer's in the news, you might have heard a lot about these treatments that are starting to emerge. In the last few years, we've started to see the first positive trials within Alzheimer's, which is hugely exciting. So 2 drugs called, and let's see if I can say these correctly, donanemab and lecanemab. These are antibodies that remove the amyloid from the brain. And what these historic trials show is that doing that can actually slow down the progression of disease. So we call this disease-modifying therapies, which is what we are all striving for, something that actually will change the course of a disease process. Now, this does not come without caveats. The drugs have really a very modest effect, and they are only effective at very early stages of disease. And they also come with some really quite serious side effects in certain subgroups of individuals. So this is not the end. This is not job done, but it is reason for optimism and reason to think that actually we're thinking along the right lines as we start to develop these treatments. Really excitingly, we're also making huge progress in terms of the early diagnosis of Alzheimer's disease. And anybody who is listening who has had a loved one affected by Alzheimer's will know that diagnosis is not always quick or straightforward. Now, some of you may have seen in the news in the past week a trial from my UCL colleagues, Professor Jonathan Schott and Ashvini Keshvan, where they are now testing a blood test for Alzheimer's out in kind of real world settings. So quite often we develop tests in the lab that work very well in a research setting, but of course we need to understand that these work in kind of real life clinics. And actually, this blood test is a very, very sensitive way of detecting the tau protein, which builds up in the brain. Within the blood, some of it leaks into the blood. And if we have sensitive tests to detect that, it can be an indication that somebody is in the early stages of Alzheimer's disease. So this is really an incredibly exciting time for diagnosis and also for treatments, although there is really a long way to go. And so this kind of allows me to summarize where we are now and then start to talk about our own work and where we might go in the future. So I hope I've convinced you that Alzheimer's is really an urgent public health challenge. And understanding the genetics of disease from these incredibly rare families has allowed us to start to put together the changes we see in the brain in an order. So we start to understand what happens first and what comes later on. It's been a long road to successful treatments, but the first disease-modifying treatments are now starting to show success in clinical trials. This is far from the finished kind of goal, but it does put us in this new and exciting era. And the fact that that is being accompanied by new methods for early diagnosis, I think is incredibly exciting and will have real benefit for patients. But we have to acknowledge the caveats that the effects of these drugs are modest. They're only successful in very, very early stages of disease, and they may be accompanied by side effects. And this drives within the field a need to understand the very earliest changes at the cellular level and also have a diverse range of treatments. So not focusing on this single target, actually thinking about what different targets within the disease could be beneficial to address. And so this brings me to the question that my lab is interested in, which is why do brain cells die in dementia? We see the protein pathologies, we see the loss of cells over time, but what is happening at the molecular level that I talked about right at the beginning that is causing this cell death to happen? And to address this question, we need to think about modeling dementia in the lab. So again, thinking back to the genetics of Alzheimer's disease, when we know that a gene causes Alzheimer's, this is really valuable information, but knowing the gene alone doesn't tell us how it causes Alzheimer's. To address that, we build models in the lab. So we can take the faulty gene and put that into cells or put that into animal models such as the mouse or the fruit fly. And I think the next lunch hour lecture in the series is actually all about the fruit fly and how that helps in dementia research. Now, these models have been incredibly valuable, but they are, of course, very different to a human brain. The size and complexity of the human brain and our lifespan is completely different to any of these alternative approaches. And so what we really need is a way that we can generate human brain cells in the lab and study what's going wrong in a species-specific way. And this is very challenging because I've talked about postmortem tissue. The reason we study the brain at postmortem is because we cannot access that material during life. It's not possible for us to take brain biopsies throughout somebody's disease course to see how the changes are evolving. So as I mentioned, and just to give you a kind of visual representation of that, the human brain is really vastly different in terms of the size and complexity to other organisms. So how can we address this? How can we have human brain cells to study in the lab? Well, the approach that my group and many others worldwide are taking is to use stem cells. So many of you will have heard of stem cells, but just to bring everyone into the kind of same understanding, stem cells are basically a blank slate. So it means they have the potential within them to become any cell type of the human body. And I really love this cartoon that stem cell can be anything it wants to be when it grows up. So what that means for us is we can have stem cells in the lab and we can turn them into neurons. We can turn them into other cell types of the brain. What has been incredibly transformative in the field is the fact that we are now able to make stem cells from skin cells. So skin cells, as most of you may know, are constantly renewing. Our skin cells are constantly dividing and renewing. This means we can easily grow skin cells in the lab. It's much more accessible to take a piece of someone's skin than it is a piece of someone's brain. And thanks to techniques that won the Nobel Prize actually in 2012, we are now able to take a skin cell and take it back in time to the point when it was unspecialized. We call these iPSC cells, IPS cells, because we are inducing pluripotency. And pluripotency is the scientific word for the blank slate, the potential to become any cell type. Now, what does this mean in terms of Alzheimer's research? Well, I've been really privileged to work with clinical colleagues led by Nick Fox and Natalie Ryan, that now if somebody comes to their clinic who has one of these genetic forms of dementia, we ask them to donate a small piece of skin. It's the size of kind of a rubber on top of a pencil. We grow the skin cells in the lab. We use those skin cells to make the stem cells, and then we can turn those stem cells into brain cells. And because those cells contain the person's DNA, we have a personalized patient-specific model of dementia that we can study in the lab. And just to give you, again, some visuals of what this looks like, this is one of the skin biopsies that we receive. As I mentioned, it's about the size of a rubber on top of a pencil. The skin cells grow very easily in the lab. So this is under the microscope in the bottom left. You can see the dark blob is the piece of skin, and the cells radiate out from that piece of skin. And what's incredibly nice is we can freeze these, and then when we need to work with a particular sample, we can revive them and thaw them, and the cells will start growing again. And on the bottom right of your screen, you see the kind of frozen stocks of cells that we keep. So we make the stem cells and we turn them into neurons. I've got this picture in my background because it's one that I find particularly beautiful. What you can see is the very young neurons look a little bit like firework that exploded, but the green, very delicate lines that you see radiating out are the cell's axons. So in the brain, our cells communicate by sending electrical signals to one another, and this is through these kind of wire structures that they do that. And the cells don't only look like a neuron, we can actually eavesdrop on them, and we can see them communicating with each other in the dish. And that's what you can see on the screen here. Really powerful imaging that's letting us see when the cell sends a signal to the neighboring cell. And this is really valuable to us because it means we can take cells from people with and without dementia and look if the behavior of those cells is different, and also look how the behavior of those cells changes over time. Now, the cells I've shown you so far are in 2D. We kind of grow them on layers of plastic. And of course, that's not how our brain is. And where the future is really headed are what we call organoid cultures. So these are 3D cultures of brain cells, which start to be a bit more structurally like the brain. This is an image of some of them. So each one of the little white dots you see is an individual organoid. They're about the size of a lentil. So they're very, very far away from being an intact human brain, but they start to give us a more patient-relevant model to study in the lab. And again, if we take one of these cells and we take an image through the middle of the organoid, we see something like this. So we have those same cell types, but each color here is a different type of cell, and they're organizing themselves in a spatially separate way. So this is incredibly exciting, and I told myself I was allowed one graph in my talk, and here it is. We're starting to see in the culture dish, in cells that we grow in the lab for months, not for years, just for a few months, we start to see some of the signatures of Alzheimer's disease developing. And on the left, we're starting to see higher levels of the amyloid protein, and on the right, we start to see changes to the tau protein. So this is really an opportunity for us to have an understanding of early disease in patient cells, but also to use those cells for future drug screens, which is something that I work really closely with Fiona Ducoteur, who's chairing the session, to do. So just for my final few minutes, I wanted to take a step back and actually talk about late onset Alzheimer's disease. We've really focused a lot on autosomal dominant or familial disease because that has the genetic cause that lets us gain so much insight. But many of you, as I mentioned, will know that most Alzheimer's disease, 99% of Alzheimer's disease is not genetic in the same way. So in familial Alzheimer's, we really think that the driver is altered amyloid production. But in late onset Alzheimer's, we know it's a combination of genetic risk, but also environmental and lifestyle factors. Now, the genetic risk actually hints at clearance of amyloid being something that might contribute to disease. And to understand genetic risk, there have been many studies using what we call genome-wide association approaches. And this is where you study thousands and thousands and thousands of individuals to detect genes that have very, very small effects. And this was just a cartoon I put together to try and explain that actually we all carry a certain amount of genetic variability, and some of that variability will increase our risk for certain diseases, but not to the point where it would cause us to develop a disease. It would actually increase it by less than 1%. And what you can see here is we've identified, by looking at large populations, genes which really increase the risk by a small amount of developing Alzheimer's. And these genes, we believe, enriched in a cell type called microglia, so not in neurons, but by the glial cells I mentioned at the very beginning. And this, we think, links to inflammation in the brain, but also how our brain responds to the amyloid. So rather than producing too much, we just can't respond as effectively. But the effect is the same, which it kind of tips this balance in favor of the disease pathologies. And one thing happening now that I find incredibly exciting, but it's also incredibly new, is this idea of genetic escapees. So rather than genetic risk, we're now understanding that some people have genetic resilience. So we may carry genes which actually protect us from developing Alzheimer's disease or other diseases of the brain. And although this is a very, very new area of research, it's exciting for two reasons. One, it really again tells us that inflammation is an important process that we need to understand. But two, for me, this is nature's way of showing us that a cure is possible. If we can carry genes that actually stop somebody from developing Alzheimer's, if we can understand that and mimic that process, that really gives us insight into how to treat the disease. I mentioned inflammation, and I thought I would finish my part of the science part of the talk by just saying it also tells us we need to look beyond the neuron and other cell types. And with stem cells, it's their real power that we can turn them into any cell type that we are interested in. So I talked already about neurons and organoids, and at the bottom you can see some of these glial cells that we've made in the lab. And we're really trying now to understand how these cells all work together to either increase our risk or increase our resilience of developing Alzheimer's disease. And of course, as I said at the beginning, Alzheimer's is not the only cause of dementia, and the same tools, the same technologies, and the same approaches are giving us insight into all different types of neurodegenerative disease. So just to wrap up, hopefully I've shown you that stem cells are really allowing us to build bespoke, personalized models of disease. And by combining this with genetic knowledge, we can understand the very, very earliest things that are going wrong in brain cells. It's become really clear that although neurons are the cell type which dies in the disease, we won't understand the disease by only focusing on those cells. We need to look beyond the neuron and all the cell types in the brain as a whole, and indeed beyond the brain to understand, for example, how cardiovascular health may influence dementia risk. My own work is in the cell biology of dementia, but of course it's happening in parallel with progress in early diagnosis, treatments, and a better understanding of brain health and risk. And I think although the facts and figures I presented at the very beginning are quite daunting and a little bit scary, I think we should really be optimistic about the future of dementia research. And I'm really excited to be at the place which has the clinical and the preclinical research together. And none of what we do would be possible without the participation of patients and their families. Looking again to the future, we're also excited. I showed you the slightly old concrete building at the beginning of the talk, and in 2 years' time, we're moving to a new translational neuroscience facility, which will have patients and labs to get clinical patient-facing research, so clinics, and laboratory research together under one roof. And for anyone who is London-based, you can see the building kind of emerging right now on Gray's Inn Road, and we're very, very excited about how this will accelerate understanding and treatments. So I just finished there by quickly thanking my lab, past and present, and collaborators around UCL, but also internationally. This is a big problem, and it really needs us to address it as a team sport. And I think it's we're fortunate that the field is hugely collaborative and supported by some wonderful funders. And as you may have heard, it's World Alzheimer's Month. It's World Alzheimer's Day on the 21st. And UCL has been doing a whole campaign around this. And I would really just like to leave this on your screen for 5 seconds where you can follow the link and just understand a little bit more about research happening across UCL, particularly with the use of AI in Alzheimer's research, which is incredibly exciting. So thank you so much for joining us this lunchtime. And that's the end of my talk. And I'm really excited and happy to take your questions. I will stop sharing my screen so I can see what's happening. Thank you.
Thank you, Selina, for a wonderful talk. I think we're going to jump in now to the questions. I think just to say you've really laid out in, such an elegant way how far we've come as a field and, the real hope for the future in the research, particularly that we have happening at UCL. So just as a reminder to the audience, you can submit questions on Slido and the hashtag is WAM, but we've already got questions coming in, so I'm going to jump straight into the Q&A right now. So Selina, number one, researchers around the world are investigating possible treatments for Alzheimer's disease, and some of them have very promising results. How can UK researchers develop more partnerships with others across the globe doing this type of research?
Yeah, this is a really good question. And I think I would answer that first of all by saying that Alzheimer's is incredibly collaborative and we don't operate in silos. So there is actually a huge amount of international research and collaboration already happening. Thinking, just referring again to the genetic cases of Alzheimer's that I talked about, this is really rare. So to be able to, for example, run a trial in those families, it's only possible if we collaborate internationally, because otherwise there wouldn't be enough people to enable that. The same is true of the genetics I mentioned at the end. So the genome-wide association studies that need thousands and thousands of people to have sufficient power to be able to detect these small changes. And again, that can only happen with international collaboration. So there are a number of consortiums, whether this is for trials, for genetics, for even the cell modeling that I do, that are international, and they are set up to enable sharing of data, sharing of samples, and sharing of knowledge to kind of accelerate what we do. So that international collaboration is already happening. Doesn't mean it can't be better. And I think, what a lot of this comes down to, and I'm sure many people will think this is quite a predictable answer, but it's the truth, is to have funding schemes which are specifically designed to increase collaboration and bolster those international networks. I think the enthusiasm from the researchers is there to make it happen. We just need the resources to facilitate that.
Yeah, great. Thank you, Selina. And actually a follow-on question, I guess, from the non-research side. So do you need more volunteers to help with research? So I guess a question about how can the public get involved and contribute to research?
Thank you. It's really an important question, and I'm happy to have the chance to highlight what I think is a wonderful website called Join Dementia Research. If you put Join Dementia Research into Google, it will bring up the website where you can register your interest about participating in research. And what I think is really beautiful about this website is you can set your level of participation. So some of the studies that we have, for example, can be quite involved. They can involve visits to a research centre. They can involve having blood tests or spinal tap, CSF, or brain scans. But not everyone has time for that, right? And there are other studies where it might be participation in an online interview or online questionnaire. So please don't be put off by thinking, oh, I'd love to contribute, but I don't have the time. Go and have a look, because there were studies that are open to everyone. And the other thing I would say is it's not only about people who are living with dementia participating. We also need healthy control participants. And quite often there's also research that will involve people who've cared for a loved one. So if you have any interest at all, please go and check out Join Dementia Research.
Thank you, Selina. Okay, so there's a lot of questions coming through about risk of dementia and how we can, use the emerging research to reduce our own risk of dementia. So would you be able to give us some thoughts about anything we can do to reduce risk of dementia?
Absolutely. And there's some beautiful work that has come out actually from UCL and other places about understanding modifiable risks. this is, you know, in the way that we know if you stop smoking, it reduces your risk of lung cancer, are the similar steps that we can take in our day-to-day lives that might reduce our own risk. So we know, for example, that managing blood pressure in midlife will reduce risk of dementia later on. Essentially, much of the advice will overlap with what we're told for cardiovascular health. So healthy diet, regular exercise, don't smoke, low alcohol consumption, these will all maintain brain health and reduce risk later on. We also know there are other environmental risk factors that emerge, things like pollution, where we think there could be a link, but we maybe don't fully understand it. But yeah, certainly what's good for the heart is good for the head.
No, it's true. So moving from risk to sorry, there's so many questions. Thank you all for
I'm trying to
submitting
give
them.
quick answers as well. I'm not always brief, so I'm trying.
Yeah, so actually thinking about moving from risk to signs of dementia. So there's a lot of questions coming in around, you know, are there ways to pick signs up early of changes that could be driving dementia, and also whether AI and wearable devices might be able to be applicable in that space. So that's a good meaty question for you there, Selina. Good luck.
Yeah, really good question. And I kind of want to start with a disclaimer, which is I am not a medical doctor. I'm not clinically qualified. But from what I understand from talking to colleagues, but also talking, you know, to friends who've had loved ones with dementia, quite often the early signs or changes are noticed by people in the family rather than the person themselves. And I think if you have any signs which you may be concerned about, what I would recommend is to actually talk to your GP, talk to the person who's affected, if it's not you, of course, and maybe encourage them to go and have a conversation with the GP. But also to reassure people, I mean, something that when I'm giving these talks in person, nearly always somebody will come up to me afterwards and say, my memory's really got a lot worse. And, you know, most of us will have memory troubles at some point in our life. If we're busy, if we're stressed, as we get older, our memory will get worse, and that's not necessarily anything to be concerned about. But if you do have any worries, I would, you know, go in and speak to your GP. For the use of AI, I'd signpost back to the slide I put at the end, which will talk about, lead you to links about AI being used in research across UCL. It's an early time, but I think incredibly exciting. One way that we are thinking about using AI in our own research, for example, I've shown this picture many, many times, and I said that, you know, we're comparing cells from people with dementia from cells from people without dementia. Now, AI and machine learning may help us to analyze those images and detect changes that may be too subtle for us to pick up with existing means. So I think it will really increase the power of our research. And of course, I'm talking about cells in a dish, but we can use those same approaches to looking at brain scans, for example, looking at big data sets to look for patterns that we may have not detected by more conventional means. So I think although it's early days, AI has huge potential to kind of help us in dementia research.
Thanks, Selina. So I'm going to move on now to some questions around treatments. Obviously, you gave a really nice introduction to the emerging treatments in recent years, and our audience are asking about the future of research and what other promising new treatments are coming through the pipeline, and you know where they are, and if you could comment on that.
I'm going to give my answer, but Fiona, I might invite you to chip in on this. Seeing as your job is drug discovery, you might be better placed than me to answer it. But just to, I guess, to give my own perspective, I mentioned right at the beginning, it's important we don't focus on a single target. And one thing that I've been really excited about and didn't have time to talk about are treatments which are actually more looking at targeting the tau protein rather than the amyloid. So although we know the amyloid is upstream and it seems to happen first, there is good data to suggest that the tau is more closely linked to the degeneration of neurons. So I think tau as a target is incredibly exciting, and there are a number of trials that are, well, many trials that are addressing tau. One that I think is really exciting is a tau antibody that I saw yesterday. One of the pharmaceutical companies are just given kind of fast track status to try and kind of make those trials happen quicker. And there are also some genetic therapies that are trying to reduce the levels of tau by targeting the tau gene that so far have only reported safety data. But I think they look incredibly exciting. And of course, the third thing that I hinted at the end, but I didn't have a chance to go into any detail, is about the role of neuroinflammation. And actually, if we can, I mentioned that in some cases of Alzheimer's, a contributing factor could be that when the amyloid appears, our brains don't respond to that amyloid very effectively to get rid of it. But maybe we can boost that response and help the brains kind of deal with the harmful proteins. So I think that's a kind of really exciting area of treatment. But Fiona, I welcome you to correct me or add to that.
No, I 100% agree with you. I think, you know, I always think about that umbrella slide that you showed at the very beginning about the different causes of treatment that manifest as very similar symptoms in patients. And I think that that's exactly the reason we need to go after lots of different disease mechanisms with treatment. So, and also have ways to identify patients with certain underlying causes that we know which treatments will work in them. So there's a lot of research across that space. And then it leads it perfectly into the next question, actually, Selina, which is very much directly linked to your technologies and expertise. So this is a question: can stem cells be used as a treatment as well as a research tool?
It's a super question. And my answer is that for Alzheimer's, I think cell replacement by using stem cells or stem cell derived neurons would be incredibly difficult. Because if you think about the slide I showed you of the pathology of Alzheimer's, the loss of cells has been really widespread throughout the entire brain. And it's not a case of simply just putting those cells back. We need to give the right cell type and they need to connect in the right way. And you know, the different regions of our brain have many, many different types of neurons. So I think it would be quite difficult. I would never say never to any of these things. However, having said that, I would highlight two things. First of all, there is a trial for stem cells to be used as a cell replacement therapy in Parkinson's disease, which is different to Alzheimer's, but shares some commonalities. The reason that's maybe a little bit more, I hesitate to use the word straightforward because nothing is straightforward, but in Parkinson's disease, we know it's a much more restricted area of the brain that's initially involved. So you could think about replacing those cells. Maybe it would be a little bit more straightforward than in Alzheimer's. But secondly, the other thing I wanted to say is cell replacement has been tried in mouse models of disease, not work by us, but by others in the field. And they were able to use that to identify certain molecules, growth factors that support brain health, that they could then take forward to a human trial. So not the cells themselves, but the cells giving the information about what molecules might actually be effective as treatments.
Fantastic. So I'm going to ask you one quick question just to wrap this up, because we're coming towards the end of the session. So regarding the patient participants, as in those who donate skin biopsies to turn into stem cells, do you keep in touch with them about the findings, or how do you communicate with people who contribute to your research?
It's really an important question, and I think we have to make the balance of, if somebody is generous enough to selflessly give samples for research, I believe they have a right to know what we're doing with them and what the findings are. But of course, we have to treat the samples anonymously as well, so I can't give individual feedback. But what I do is we have a number of kind of patient and participant support groups around the institute, and either me or someone from the team goes every year to give an update about what we're doing as a lab, what progress we've made, and what those cells are being used for. And it's something that I love doing. I hope I will do it for many years to come.
Well, thank you, Selina. And unfortunately, we've run out of time. This is a topic we could have spoken about all day, I think. So thank you once again for a great presentation. I just want to let the audience know that you're going to see a QR code for feedback about the session, and give you a reminder that our next UCL lunch, our lecture on unraveling dementia, will be on the 2nd of October 2025 at 1 till 2 p.m. And thanks again for joining us, and I'm now going to close the event.